You know, when we think about radiation, we usually think in really simple terms. More dose equals more damage. But what if I told you that's not the whole story? It turns out the type of radiation can be just as important, if not more important, than the amount. So let's dive into this fascinating riddle, shall we? Okay, so let's kick things off with a question. Say you get a 1 gray dose of X-rays and then a 1 gray dose of neutrons. Is the biological effect the same? I mean, it seems totally logical, right? Same dose, same damage. But the surprising answer, and really the whole point of this explainer today, is a big, fat no. 1 gray of neutrons causes way more biological damage than 1 gray of X-rays. And the key to unlocking this whole mystery is all about how that energy gets deposited at a microscopic level. All right, so here's our game plan. We're going to tackle the radiation puzzle, figure out how scientists actually measure biological effectiveness, then see how dose and fractions totally change the game. From there, we'll dig into the deeper cause, something called LET, and finally, we'll get to that aha moment, the DNA sweet spot. Okay, so if 1 gray isn't always the same as another gray, how on earth do we compare different types of radiation? Well, scientists needed a tool, a way to standardize things. And that tool is called Relative Biological Effectiveness, or RBE for short. Now, the definition sounds a little technical, but it's actually pretty simple when you break it down. RBE is all about comparing some test radiation, like neutrons or alpha particles, to a universal standard, which is usually a specific type of X-ray. It's just a ratio. You take the dose of X-rays needed to cause an effect, and you divide it by the dose of your test radiation needed to cause that exact same effect. Okay, to make this real, let's walk through a classic experiment. Picture this. You've got a bunch of plant seedlings, you hit some of them with different doses of standard X-rays, and you hit other groups with different doses of, say, fast neutrons. Then you just watch and see what dose it takes to kill off half the seedlings in each group. That's called the LD50. And boom, this is where it all clicks. Let's say it takes 6 grays of X-rays to hit that 50% kill rate, but it only takes 4 grays of neutrons to do the same job. You just do the simple math. 6 divided by 4 gives you an RBE of 1.5. That literally means that for killing these seedlings, the neutrons are 1.5 times more biologically damaging than the X-rays. Sounds simple enough, right? Well, hold on, because it's about to get a whole lot more complicated. Yeah, this is where the plot really thickens. Because it turns out that RBE isn't some fixed constant number. Nope. It can change, and change dramatically, depending on how you deliver that dose. Okay, take a look at these two graphs. On the left, that's panel A, you're seeing what a single big dose does. But now look at the right, panel B. This is what happens when you take that same total dose, but break it up into 4 smaller chunks, or fractions. Just look at the difference between the X-ray and neutron curves in both situations. Something really big is happening here. And the numbers here are just wild. Remember our original RBE? It was 1.5 for a single shot of radiation. But when you look at what it takes to get the same effect with a fractionated dose, the RBE for neutrons skyrockets all the way to 2.6. They become so much more effective, relatively speaking, when the dose is split up. So what is going on here? Why does this happen? The whole secret is something called sublethal damage repair. And this quote really nails it. For X-rays, that curve has a shoulder, a flat part at the beginning, and that represents damage the cell can actually fix. So every time you give a new fraction, a chunk of that dose is basically wasted just getting past that repair mechanism all over again. So here's the bottom line. Think of it like this. X-rays are like a boxer throwing lots of jabs. The cell can take a few, recover between rounds, and you have to keep jabbing just to wear it down. But neutrons? They're like a knockout punch. The cell has almost no ability to repair that kind of damage. Their curve has a tiny little shoulder, so almost no dose is wasted. The more fractions you use, the more inefficient the X-rays get, which makes the super efficient neutrons look even more powerful by comparison. Okay, so this huge difference in the kind of damage, that brings us to the deep down physics of what's really happening. We need to talk about something called linear energy transfer, or LET. LET is, a fancy term for a really simple idea. It's just the density of the energy hits. Think of low LET radiation, like X-rays, as a shotgun blast from far away. The pellets are sparse and scattered. High LET radiation, like an alpha particle, is more like a cannonball, just plowing through and depositing a ton of energy in a very tight, dense line. And this graph just lays it all out perfectly. Just watch what happens.
As you go from the low LET X-rays up to the neutrons and then to the super high LET alpha particles, that survival curve just gets steeper and steeper. And look, that little repair shoulder, it just vanishes. The higher the LET, the more effective the cell killing.
But, and this is the biggest twist in the whole story, check this out. If you plot that biological effectiveness, the RBE, against the LET, something truly bizarre happens. It doesn't just keep going up and up. The RBE climbs, hits this incredibly sharp peak right around 100 kiloelectron volts per micrometer, and then it starts to drop off. Whoa. So there's a most effective level of LET.
And that leads us to our grand finale, the aha moment where all the physics we've been talking about slams right into the biology of our own DNA. So you gotta be asking, why? Why that specific number? What is so magical about 100 keV per micrometer?
The answer? It's all right here. It's so elegant. It all comes down to the #1 target for radiation inside a cell, DNA. That curve on the bottom shows the RBE peaking, and the picture above it tells you exactly why.
Here it is. This is the whole key. At that magical LET of 100 kiloelectron volts per micrometer, the average spacing between the little energy explosions is 2 nanometers. And guess what else is exactly 2 nanometers wide? The DNA double helix. It's a perfect match. A single track of radiation at this LET has the highest possible chance of breaking both strands of the DNA at once, creating a double-strand break, which is the deadliest injury a cell can suffer. So it's a total Goldilocks situation, right? The low LET stuff, like X-rays, is too spread out. It's not likely to hit both strands. The super high LET stuff, that's just overkill. You're wasting energy on a target that's already obliterated. But that middle one, the optimal LET, it's just right. Perfectly tuned to destroy DNA with maximum efficiency.
And that brings us right back to the real world with a huge, powerful question. Now that we know the precise physical key to causing the most damage to a cell, how do we use that? How do we build cancer therapies that are perfectly tuned to hit this DNA sweet spot, to be absolute assassins for tumor cells while leaving healthy tissue alone? I mean, the answer to that could change everything.